I'm particularly interested in trying to convert some of these epigenetic targets for drug discovery. And I'll share with you something that I worry about. I'll discuss, first of all, some of the things that we've been doing, what we've started doing, and I think what we're going to have to do more and more of in the future. So the title I've given this is a pre-competitive partnership. So it's a public-private partnership to accelerate functional studies with epigenetic proteins. So we're generating small molecule inhibitors for these proteins, and we're looking to see what they do in disease systems or cellular systems. Basically because we want to try and discover pioneer medicine. So these are medicines working via completely novel targets. So my first slide, so what does the SGC do? Well, what we do is we work on new human proteins. We don't work on rodent proteins, we only work on human proteins. And for those proteins, we generate new target validation tools. So tools that you can use to validate targets, which will hopefully go into industry for drug discovery. So we generate the purified human proteins, we generate assays for those proteins, we generate small molecule inhibitors, antibodies, the three-dimensional X-ray structure of those proteins. But importantly, what we do is everything we generate, we make it freely available. We give it to any academic, any biotech, any pharma, because we believe that's the best thing we can do to facilitate science and therefore facilitate drug discovery. As a consequence, we've become a hub for innovation because we only work on new proteins and we generate these new tools. We've also become a hub for pharma. So we now have 9 large pharmaceutical companies that are working very closely with us, and each of them has contributed $8 million to our effort. So potentially we have $72 million of private resource. We've also become a hub for academia. So you can imagine every academic who comes into my office wants to work with us because they appreciate that we'll share all of our reagents, we'll share all of our know-how, all of our expertise, and that transparency creates a lot of trust, which is great for collaboration, it's great for science, and it's great for drug discovery. This is a way essentially of pooling resources, sharing risk in drug discovery, and it's trying to crowdsource early discovery. Our focus, we work on families of proteins. These are the protein families that we work on. I'll share with you some data that we've generated in epigenetics. A lot of the work we do is in sort of cancer assays, but we're trying to do more and more in inflammation, and I'm trying to push more and more into neuroscience, because one of the reasons for working on these proteins is I think they are going to be better targets for drug discovery than many of the targets that industry has traditionally worked on. So let's just share with you some data. So I said we work on families of proteins. So one family of proteins we work on is the broma domain. Now in humans, there are 61 of these domains, 61 of these proteins, and they're shown here by this tree. So 4 years ago, we generated this inhibitor, which we call JQ1. It exists as 2 enantiomers. This is the active inhibitor, and this is the inactive. And the active inhibitor is potent at this subfamily of broma domains and has little or no activity at any of these other proteins. We worked with a colleague, Jay Bradner at Harvard, who has access to patient material, and in particular a rare type of cancer called nut midline carcinoma. So he put the compound on to these patient-derived cells and did a simple proliferation assay. So this is the vehicle in black and the compound in red. You can see the compound reduces proliferation. In another patient-derived cell line, another nut midline carcinoma, again, it reduces proliferation. He showed that it also increases apoptosis, and then Jay actually took this into a xenograft model to see what it does to tumor size and tumor volume. So this is the tumor volume, and this is days along here, and you can see with the vehicle, this xenograft, so that this is nut midline carcinoma cells from patients in mice. You can see that the tumor gets bigger and bigger. You give the compound, and it stops the tumour from growing. I should say at this point, Jay Bradner wanted to take this compound into patients because he knows that anybody that's diagnosed with nut midline carcinoma, within 3 to 6 months they're dead. There's nothing out there that works. Let me just share with you what's happened since. So we generated this probe, this molecule, and we've given it away to several labs. But since then, that molecule has been looked at in a whole range of different types of cancer. And there's positive activity. Last year, we published with Jay a paper suggesting that this target, this BET inhibitor, could be a new target for male contraception. So this was a paper, it came out in Cell. It was all, I think, completely hyped up in the US and in the UK and in China. And I'll come to why I'm sharing this data with you in a moment. And then earlier on this year, there was another paper in Cell with the same compound, now showing it reduces cardiac hypertrophy. So I've shown you now a new target, a new inhibitor. I've shown you data in cancer. I've shown you data as a male contraceptive, and now data in cardiac hypertrophy. And then GSK generated a similar compound called IBET, so it inhibits the same subfamily of proteins, and they looked at it in a model of septic shock. So if you give LPS to mice, lipopolysaccharide to mice, then after 40 hours all the animals are dead. But if you give this compound either an hour before, so here, or an hour and a half afterwards, all the animals survive. So this target may be useful in septic shock. So here's a new target, 4 different indications. And I think this illustrates the challenge we have in deciding where we're going to take these targets. This is the challenge that industry has in deciding what targets to work on and which patients to take them into. Let me just share with you what the impact of this one inhibitor has been. So we published it in Nature in December 2010, and by middle of this year, that paper had been cited more than 200 times. As a consequence of that one paper, there was, by then, a further 172 papers on this family of proteins and disease and so on and so forth. We've given that molecule out to more than 300 labs across the world, and all these labs are profiling it in various types of cancer or inflammation or infection or whatever. And you can imagine the breadth and depth of biology that's being enabled by this novel inhibitor. Now, there's no way that GSK or Pfizer could afford to do that. We can afford to do it because we don't pay the academics anything to do those experiments. Many of our pharma partners have started proprietary efforts, and Jay Bradner managed to secure $15 million of VC funding to set up a biotech called Tensure to take that target into the clinic. So this is one pre-competitive inhibitor, and that's been the impact on science and on drug discovery. Let me share with you another one. We work on another family of proteins in Oxford called the demethylases. And we worked with GSK to generate this molecule, which is an inhibitor of this Jumonji demethylase, JMJD3. And you can see that it's potent and it's selective over these other demethylases shown here. And this is just to show here, these are cells that are transiently transfected with the demethylase. And you can see these cells that express the demethylase do not express the trimethyl mark on lysine 27. Whereas in the presence of the inhibitor, these 4 cells that express the demethylase, you can see all of them are expressing that mark. So we put this compound onto breast cancer cells, and it causes apoptosis. And now, since then, there's been data in a number of other types of cancer that this molecule could be useful. We've also put this onto human macrophages. So these macrophages are throwing out cytokines. One of them is tumour necrosis factor. And you're all aware that anti-TNFs are currently used in the clinic to treat rheumatoid arthritis and inflammatory bowel disease. By the way, they're selling $25 billion a year. This compound causes a dose-related reduction in the production of TNF from human macrophages. We then put it onto osteoclasts and basically showed that the same compound reduces bone resorption. So a similar story. New target, new molecule, data in cancer, data in inflammation, data in bone. I would say that I don't think there is anybody on the planet who can tell me where that target will end up. It may be something to do with cancer, it may be something to do with inflammation, it may be something to do with bone, or it could be something completely different. And this is the challenge we have in target validation and drug discovery. We've now generated a pipeline of these inhibitors. So when we start on these new epigenetic proteins, we have to develop assays, cell assays, and ultimately get cell activity. We start with no molecule and then build in potency and selectivity until we get these inhibitors. Now, all these inhibitors are freely available. This slide is out of date. There should now be another couple on there as well. And they're freely available to anybody who wants them. I've just shown you data with 2 of those inhibitors. Now what we're trying to do, it's all fine getting these inhibitors and giving them away to various labs across the world, etc. But as you can appreciate, the variability in biology, the variability from lab to lab, it's very difficult to compare data in one lab sometimes with data in another. So what we need to do is to try and accelerate this science, and so what we're now going to do is to set up a platform of human cells and screen our inhibitors in that platform of cells. So we've already started doing this with a company called DiscoverX based in San Francisco. So what they have is a panel of human cell lines, so endothelial, fibroblast, epithelial, etc. What they've already done is they've taken 3,000 of the existing drugs and profiled them on this panel of cell lines, and they've come up with a compound, a phenotypic signature for each of those compounds. And what they've now done is they've taken some of our new inhibitors and profiled them in their panel and provided us with that data for free, which is great, because these are pretty expensive assays to run. But they've given us this data. And so this is a new inhibitor, a CREB-BP inhibitor. And the data is suggesting that the molecule is anti-proliferative, potentially immunomodulatory. There's also data suggesting effects on matrix remodeling and also effects on angiogenesis. And they can put these compounds on different doses, so these different colored lines are a dose response, essentially. They've also done another inhibitor that we've generated, a BAS2B inhibitor, another bromodomain inhibitor. And you can see by comparison with the CREB inhibitor, this looks pretty inactive, but there is some hints that this compound could also be anti-proliferative as well. So this is the sort of data we're generating now in cell lines, but I think that's not going to be good enough. So what we're now intending to do is to screen these compounds in a medium throughput format in a platform like this, but using high content readouts, but using primary human cells. And that's what we're now setting up in Oxford and in Toronto to allow us to profile these probes. But that's fine. Putting them into cell lines is great. But ultimately, I think target validation happens in patients. So I'll share with you one of the things that we're now trying to do with Paul Lasco's help here. So frankly, pioneer drug discovery. So this is coming up with completely new targets. I'm not interested in another tricyclic antidepressant or another non-steroidal anti-inflammatory drug. Completely new targets. That's what patients need. Frankly, drug discovery is a lottery. When we take pioneer targets into phase 2, so this is the first time we test them in patients, the failure rate's more than 90%. So can you imagine, you spend 5-6 years of resource, you go into patients for the first time, more than 9 times out of 10 it fails. It's a waste of money, it's a waste of people's careers. And actually, when you think that there's probably 20 companies doing the same target in parallel in secret, it's also a horrendous waste of patience. So, frankly, we're not good at target validation. That's why we fail so many times. Target validation occurs in patients. It doesn't occur in animals, and I'm afraid, as much as I may like it, it doesn't occur in cells. And I've made this comment, many academics and many pharma groups work on the same targets in competition. And this duplication is just wasteful, inefficient, etc. So what we're intending to do is to set up another public-private partnership. Now, we're not just going to generate these reagents, but what we intend to do is to generate clinical molecules and take them into patients, again, completely in the absence of IP, making everything freely available. We're going to treat it as a knowledge creation endeavor. That's why we're failing 9 times out of 10, because we don't understand human disease. We'll access academics quickly and freely, just like we are in the SGC. The targets we work on will be selected by our partners and some of our academic collaborators. The patient groups and the regulators are saying they want to be actively involved in this. And of course, then industry, what they will do is they will create proprietary molecules on those 1 in 10 targets that are likely to show some activity. So the intention is to take new targets, some of our epigenetic targets, all the way through the drug discovery pipeline to do what we call proof of clinical mechanism in a phase 2 study in patients. In the process, we will generate all of these reagents in blue, and we'll make those freely available. We'll use those reagents to produce these publications here in red. And these reagents and publications, they'll facilitate more collaboration, more leverage funds, and they'll crowdsource more of these proof of clinical mechanism studies. The patient groups are saying, look, we're desperate for new treatments. In Alzheimer's, I was at a meeting in New York last week. You know, there's been 13 failed phase 3 clinical studies in the past decade. There's 1 new Alzheimer's patient diagnosed every 4 seconds, and frankly, we're not even close to having a treatment for Alzheimer's. And this is one disease that on its own will financially cripple many societies within the next 20 years. So patient groups are saying they will help us recruit patients and we don't have to pay them. So the studies could be cheaper and faster. The regulators are saying they'll help us design clinical studies, they'll help us validate new biomarkers, they'll help us pave the path for new targets. This is a totally new way of working with the regulators. GSK can't do this, Pfizer can't do it. Where we've got to, we've initiated this project in cancer with Cancer Research UK. We're currently writing a business plan to do the same in neuropsychiatry, and CIHR is helping drive this with a number of farmers, and Paul has been particularly visionary on this. We've got discussions ongoing around doing this in inflammatory bowel disease with the Helmsley Trust and with some of these investigators. My final slide. Epigenetic targets, there's no doubt in my mind they have broad therapeutic potential. That's why we're working on them. I think they're going to be great targets for cancer, inflammatory diseases, neuropsychiatric diseases, metabolic diseases. But frankly, too many targets are potentially useful for cancer. I mean, there's not many targets that are not showing some activity in some of these cancer assays. This is exactly what happened in the industry 10, 15 years ago with kinases. Every kinase they tried worked in some sort of cancer. We're now starting to triage these targets in patient cells. But pioneer drug discovery, it's too high risk, it's too expensive, and it takes too long. There's too much duplicative activity on targets destined for failure. I think the only way forward is to pool public and private resources, the way we're doing in the SGC, access global academia, work with patient groups and regulators to de-risk some of these pioneer targets. And ladies and gentlemen, I think we have to do it pre-competitively. If we don't, in 20 years' time, we will not have a new treatment for Alzheimer's. Thank you very much.
All right, thank you very much. We have time for a few questions here in the front.
Of this idea are, first, what do you mean by de-risk targets? And second question, what are the regulatory aspects? You had very briefly a slide of what regulators would do. What are the barriers and what would they do?
So by de-risk, I mean when we take 10 targets into patients, new targets for the first time, 9 of them will fail. 1 of them will show some activity. So that one target is de-risked. It's likely to show some activity in patients. So what we're saying is that industry should start working on that 1 in 10 instead of competing on the 9 in 10, which are essentially rubbish, etc. So what the regulators are saying, of course, because this is a knowledge creation endeavor, this isn't about me and Paul or anybody else trying to make money. This is about doing what's right for patients. It's saving patients, it's saving duplication, etc., etc. So what they're saying, of course, you know, they have access to data on every clinical study that's ever been done in Alzheimer's or pain or whatever. But they can't share all of that knowledge and data with you guys. You know, they can't publish it. But they have that know-how. They know what targets are likely to work and what targets are not likely to work, what we've learned from previous studies in aggregate. So what they're saying is that if we do this, they will be active participants. They'll actually help us design what the phase 2 study should look like, how we stratify the patients, how we dose the compound, what biomarker we use, that sort of stuff.
So many targets, at least in cancer, the mutations are heterogeneous, or one allele, not the other, suggesting so there's still a wild-type copy of that modifier present. Is the Structural Genomics Consortium looking at any other modulators? In other words, not just inhibitors. What about trying to activate the remaining allele?
No, we're not doing that. We're not looking at modulators. We're largely focusing on inhibitors at the moment. As you appreciate, there's 400 proteins, there's a number of families. We're trying to get inhibitors for as many of these as quickly as we can. And I think, frankly, that the reason we've made the progress we have, that we've now got 21 of these inhibitors, and these are high-quality inhibitors, because we screen them against lots of other proteins, etc. The reason we've made such progress is that we are working closely, and I mean very closely, with 9 large pharmaceutical companies. So we have access to their medicinal chemistry expertise, we have access to their sort of compound collections and fragment libraries, etc., etc. So that's been a real help. But modulators, that'll be something for the future, I think.
Okay, so if somebody wanted to access one of your compounds, would they just email you, or is there a procedure in place for setting up a collaboration?
There's a website, just go there, ask for it, drop me a note, I'll connect you up with the right people.
Excellent. Thank you. All right.